Hi, Mary Misako Axhelm here. Hi. I'm working on lab 3A. I call it 3A. It's the second part of the third week lab. And it's the Bacterial, Morphology, and Staining Techniques lab. I just completed doing the first, I think it was, the first exercise, at least the first part of the first exercise, and that was taking the swab and putting it in my mouth and teeth and gums, which I haven't brushed my teeth since this morning, and I've had a lot to eat, so Lord knows what that could be like. Anyway, so I put it on the slide, the One Direct M, and I guess you can see the purple there against my mask. I went ahead and I put the heat on it to kind of cure it to, I don't know if that's what you call it, but that's how I do in the craft world, to make it stay on the glass slide. And then used the Crystal Violet Gram stain over it, and then rinsed it off till it went clear, and very lightly dried it, and then looked under a microscope under the low, or the scan, the medium, or the scan, the low, and the high, and I was able to see it in those three different views and magnifications, I should say. So that was really interesting. Now I'm moving on to the 3 neg M, and so I'm going to take off my mask because I have to use the swab, and I'm almost thinking I want to take off the goggles too. So I'm going to get a hold of the swab and put it on my mouth again, inside where my teeth and gums are, and try to get a sample. So I've already placed, let me just go ahead and put this way so you guys can see. All right, so I've already placed the drop here of the Congo Red on the slide, just underneath the 3 neg M. And I'm checking this off as I go, actually. So now I'm taking the sterile swab, the second one, and I'm going to scrape the inside of my teeth and gums again, which I'm doing right now. So I'll do the bottom part of my mouth this time. Okay. And it says to place a swab onto the drop of Congo Red stain and agitate the sample to transfer cells from the swab to the stain. Okay, so I'm going to agitate it. Hopefully this is working. I don't know. I don't even see any more red stain anymore. It's kind of absorbing it. I'm not sure if I was supposed to do it like this, but there it is. All right, so now you can't even see the stain. All right, it says to take another slide, like a clean blank one, and then put it at a 30-degree angle to contact and spread the dye across the slide. Okay, I'm gonna do that, but there really isn't any dye. So now I'm wondering if I need to add more dye. Maybe I will do that. So I'm gonna add one more drop. It said just to put one, but I'm gonna do one more because it kind of got rid of it with the Q-tips, so I'm not exactly sure what happened here, but I don't know. I don't know what I'm doing, actually. All right, so now I'm going to slide it across, as it says, like a 30-degree angle. Okay, hopefully that did it. Still some on this slide, so I kind of wanted to, there we go. I hope I'm doing this right. All right, then I'm gonna go ahead and let that air dry. I'm actually gonna bring it back a little bit just so that there's more, because I'm not even seeing anything, to be honest. So there. So there. Never know. I don't know. Often it doesn't tell you where to put stuff, so I'm kind of not sure or clear where to put something. Anyway, so I'm gonna let that air dry, and I'm going to look at it under the microscope.